A very good morning to all of you. I welcome to the third day of this Atal Fdp program. And today's speaker is Dr. Archana Tiwari. She is Associate Professor in MIT University, Noida. And it's my pleasure to welcome her because she has done PhD under my supervision. So I feel proud actually to introduce her. And nowadays she has already established her research laboratory working in the area of diatom physiology. She has a good experience working with the cyanobacteria. Not only that, she has written a book and she has published more than 40 research articles in an international journal of repute. So, with this short introduction, I welcome Dr. Archana Tiwari to start her presentation. Hello, Archana.
Good morning, ma'am. Thank you so
Good
much.
morning.
Good
You
morning,
please
ma'am.
start. Good morning. Good morning. You please share your slides and start your presentation.
Yes, ma'am.
Dear attendees, I have shared the lectures which has been presented and discussed in last 2 days, but I will check whether it has been sent properly or not because it was sent through the Google Drive. I'll first download and check and let you confirm. If you have any problem, we will resolve it in the meantime.
Oh,
Please proceed.
ma'am, there's an issue sharing the window because my presentation is open on my
Okay.
desktop, and
Okay.
when I'm clicking the share button,
Mm-hmm. Okay. So let me download your presentation and share.
yeah, ma'am. Just a minute. I'll share the complete PPT with you.
Okay. Please do it.
Is my screen visible?
Yes. Not yet. Yes, it is visible. No, no, no. It's not. Yes, it is done. It is done.
And the slide share is clear, ma'am.
It's clear. Now you proceed ahead.
Yeah, thank you so much. Good morning, everyone. And it's indeed a great privilege to be introduced from the supervisor. So it's my honor to deliver this talk on application of direct and indirect photolysis in biohydrogen production. So the question is why there is need of exploring the alternative sources of energy. So one of the greatest causes which or the challenges which the mankind is facing is increased depletion of natural resources. Now natural resources, we have abundant of natural resources, but with the exponential increase in population there
Just a
is.
minute, Archana.
Yes, ma'am.
Actually, you are taking your lecture through the MS Team, so I would rather prefer that you go little slow
Okay, ma'am.
because because of the delay in the delivering the your words and sometimes pictures, and try to move your cursor so that participants will be able to connect with your
Okay. Yeah.
talk and your picture. Okay, thank you.
Right, ma'am. Right, ma'am. Thank you, ma'am. So, with increased depletion of natural resources, we have to think of some sustainable solutions wherein we can address the challenges that have arise due to depletion of natural resources. And in addition to it, the fossil fuels are also causing a rise in the greenhouse gases. So here in this picture, we can see CO2, CH4, and so many other greenhouse gases which are getting emitted. So, by virtue of this greenhouse gas emission, there is a tremendous change in the ecosystem. There's a tremendous change in the environment. And now, this environmental changes are gradually affecting us. So, if you see the second picture, wherein we have the global fossil fuel consumption, we have a lot of consumption related to the coal, to the consumption of different gases. So, when we compared the fossil fuel consumption initially in 1965, we can see the picture of India wherein the consumption was very, very less of fossil fuels because we had limited cars. Even when we go 20 years down the line, we can see that the roads were nearly empty, though we didn't had that good 8 lane or 10 lane roads, but still the number of vehicles were relatively very less. And now with this population and everybody owning a car or so, even in families we have 3 to 4 cars, which was not the scenario previously. So all of them is contributing towards a tremendous increase in the fossil fuel consumption. And in the third figure, we can see that United States, it's at the top, wherein we have a huge consumption of fossil fuel. And now India is also, exponentially rising in the fossil fuel consumption. So all these changes have led to a situation wherein it is very, very alarming. And this is the time, in fact, it is high time we pay attention to the environmental changes which are occurring due to global warming. One of the factors is the glaciers. We have melting of glaciers. The second aspect is a lowering in the snow cover, and all these contribute towards water table. So even if we have a slight change in the first parameter, say glaciers, if there is a change in the amount or the proportion of glaciers which are present on Earth, that will lead to a tremendous change in the whole water system. So one each of the factor is significant ecologically. So in addition to it, due to environmental changes, we have a lot of climatic changes. Spring is coming earlier. We have a lot of temperature fluctuations. It's a normal phenomena that we get so much of rain in winters, which was not the scenario earlier. So all these environmental changes are contributed by virtue of the greenhouse gases and the global warming which is occurring. The temperature of the land is also increasing. We have relative increase in humidity. uh, the sea surface temperature is rising. It sounds very simple, that okay, fine, the sea surface temperature is increasing, but if we imagine the situation in terms of the organism which are inhabiting the sea surface, they do have an adaptability to a certain range. So if there is a drastic increase in temperature, the aquatic system, the whole equilibrium is going to collapse, and will not have the synergy which was there in the aquatic system, which was self sustainable. So that ways, even one slight factor is contributing towards a huge devastation in the ecosystem. And we all have to maintain this uh, synergy in the ecosystem. The solution is it has to be envisaged very wisely. Here we can see the primary energy consumption from the renewable sources, and we can see that this least exploitation of renewable energy sources, while Brazil is doing a very significant work wherein they are shifting from non-renewable sources to renewable sources, which is a great step towards sustainability. Even United States has started utilizing different resources to focus on the renewable energy sources. If we compare the per capita energy consumption from the renewable sources, we can see that India is way far behind the target. Countries like Sweden, Brazil, France, and Netherlands, they are paying attention to the global scenario. They are paying attention from shifting from non-renewable energy sources towards renewable energy sources. So in India, if we see the scenario regarding the utilization of renewable energy sources, we can see that hydropower is one of the most commonly used renewable energy source, but wind energy, solar energy, and other renewable sources of energy and their utilization in India is yet to be envisaged. And I think this FDP is a great initiative wherein we can foster the application of renewable sources of energy, and more people come forward towards research and utilization of renewable energy sources. Then the question is hydrogen, and why it is the future fuel, why there is so much of emphasis paid on hydrogen and its potentiality as a renewable source of energy. It is eco-friendly, clean fuel, environmentally safe. It does not release any greenhouse gases. So this can be a great paradigm shift. Utilization of hydrogen can be a great paradigm shift from global energy system towards sustainable energy system by 2020. And we had great initiative by SpiceJet in 2018, wherein the first flight from Dehradun to Delhi did occur practically by the contribution of CSIR, Indian Institute of Petroleum. That was one initiative. We need to have more and more such experiments so that one day the dream of utilizing renewable sources of energy becomes a reality, and it has the fortune to see the day light. In addition to it, when we talk of hydrogen, the higher specific energy is 148 megajoules per kg, which is quite high when we compare to that of methane or natural gas and gasoline. So this high specific energy content is the driving force which stimulates us to understand that yes, the potentiality of hydrogen needs to be envisaged as future fuel. We can see the global hydrogen demand and its various application shares. So hydrogen can find other applications as well. And in the coming years, with the hydrogen demand increasing, more and more research is required so that we can get it to the commercial platform. So, when we talk in terms of utilization of hydrogen as a source of renewable energy, it is one of the most abundant element on this universe, and it can also, lead to a zero carbon energy source and reducing greenhouse gases. It's very versatile energy carrier. So its versatility lies in its utilization in different sectors like industry, transport, heat, and electricity. In addition to it, it can also foster or enhance energy security by our exclusive dependence on fossil fuels. Now,
हेलो।
yes, ma'am.
तुम्हारी आवाज नहीं आ रही थी बीच में, भी 2 स्लाइड पीछे जाओ।
Slide पे, ma'am.
ये 2 स्लाइड पीछे थी। हाँ, हाँ, यहीं से बोलो, हाँ।
Okay, so hydrogen is considered to be the future fuel because it is environmentally safe, and there is a potential which is possessed due to specific energy content, and the specific energy content in case of hydrogen is 148 megajoules per kg, which is significantly high when we compared in terms of methane, natural gas, and gasoline. So this whole energy statistics explains the suitability, sustainability, and the significance of hydrogen as a future fuel. And utilizing hydrogen can be a great paradigm shift from global energy system GES towards sustainable energy system by the end of 2050. So hydrogen can serve a diverse variety of applications. Hence, it is termed as a versatile energy carrier, and this versatile energy carrier finds its application in various industries, in transportation, in heat, and in generating electricity. At the same time, because it doesn't emit greenhouse gases, that can be a very good, sustainable, eco-friendly approach towards environmental conservation. And the abundance of hydrogen is much more higher, which uh, which which is yet another significant factor. In addition to it, it can enhance the energy security by reducing our dependence on the different fossil fuels. So, when we talk in terms of how to produce hydrogen or what are the methods that can be utilized for hydrogen production, we find a wide array of processes wherein we can have synthesis of hydrogen. So, one of them is thermochemical processes, coal gasifications, biomass gasifications, natural gas reformations, solar thermochemical reactions, and biomass derived liquid reformation. So here in this figure we can see that the natural gases, they contribute towards 48% of production of hydrogen, which is one of the significant percentage. In addition to it, we can have hydrogen derived from electrolysis, grid electrolysis, wind power electrolysis, and PV electrolysis. In addition to it, there can be direct solar water splitting or photoelectrochemical methods for hydrogen generation. Biological routes are yet another promising options wherein we have microbial biomass conversion, or we can have photobiological conversion of hydrogen, a great step towards utilization of hydrogen as a renewable source of energy. We can see here that when the steam methane reformation by CO2 capture and storage is utilized and the feedstock is methane, the per capita cost in $1 million, it is 226.3, and the hydrogen cost comes around 2.27 US dollars per kg. And when we utilize the steam methane reformation with no CCS, then this production cost comes down to 180 $0.5 million dollars, and the hydrogen cost per dollar per kg is 2.08. In that case, dark fermentation, we can have a much reduction in the cost, and through biophotolysis, through the direct methods utilizing algae, the cost comes around 2.13 US dollars per kg. And when we utilize biophotolysis using indirect method with water splitting mediated by algae, it can come to something around 1.42 US dollars per kg. So this number is quite promising, and there's a lot of research which is focused on the techno-economical consideration and the economic perspective towards lowering down the cost, because until and unless we have lowering down the cost, it cannot be a viable commercial application. So biohydrogen. In the previous slide, we could see that there can be a drastic reduction in the cost when we utilize microorganism, particularly algae mediated photosynthetic based biohydrogen generation. So, hydrogen from renewable and economic sources like biomass or solar energy can be utilized through direct and indirect photolysis. And we have array of mechanisms through which algae and other photosynthetic bacteria can synthesize hydrogen. So this whole process of biohydrogen production, it doesn't require any method of physical extraction. It just requires the inherent property of the microorganism to utilize the substrate and form hydrogen. The only thing that we have to provide them is an optimum temperature and optimum nutrients so that they can grow and they liberate hydrogen, which can be utilized as a fuel. Biohydrogen is considered to be much better or much advantageous because of the simplicity in operation, and we get a wide range of feedstocks. So agricultural waste, food waste, dairy waste, distillery waste, all these waste can be utilized as a source of nutrition for the microorganism, which will further lead to production of hydrogen. We have a good concept of carbon neutrality, and the cost effectiveness in operation is yet another added advantage. Though the pioneering discovery of hydrogen production by fermentation and green algae Senedesmus, it was demonstrated in 1942. But all these years down the line, the techno-economical considerations and reducing the economic cost has been a major challenge faced by the researchers working in hydrogen production. So the different methods, the biological methods for biohydrogen production include fermentation and photosynthesis. So in the first picture, we can see the photo synthesis based biohydrogen production, wherein we have two significant contributors or a catalyst, nitrogenase or hydrogenase, which are playing a vital role. And this mechanism of photosynthesis is similar to that of higher plants, wherein we have in photophosphorylation involving photosystem 1 and photosystem 2. In addition to it, we also have splitting of water that leads to liberation of hydrogen. And in addition to the direct photolysis, we can have indirect photolysis, wherein initial excitation is by virtue of the solar energy, and then we have the activity of the enzymes which are mediating this photolysis. Fermentation can yet be another option of biohydrogen production, which is utilized by some algae and photosynthetic bacteria, wherein we have synthesis of hydrogen through dark fermentation and photofermentation, so wherein we have utilization of the cycle, and there's a pioneer role of hydrogenase enzyme bringing about the H2 production. So it is all the unique metabolic potential of microorganism which is bringing about this conversion, and we have production of hydrogen. So direct biophotolysis. In the direct biophotolysis, we have the production of hydrogen, and this is just mediated by photosystem 2. And in this pathway, we have water splitting activity to the photosynthetic hydrogen production. The hydrogen ions are catalyzed either by the catalyst hydrogenase or nitrogenase to produce hydrogen gas in the presence of light, and utilizing the metabolic potential of microorganism. Microbes like green algae or cyanobacteria, they are the pioneers in harvesting the solar energy to drive the splitting of water producing O2, and we have liberation of um, hydrogen. Chlamydomonas reinhardti. There's a lot of research on Chlamydomonas reinhardti. It is a model organism for all genetic based research, leading to enhanced hydrogen production. We have yet another green algae, chlorella, Senedesmus chlorococcum, and Synicocystis and Abina, which are very frequently utilized for production of hydrogen. So hydrogen production through direct photolysis involves the photosystem 2, in which we have the light driven electron transfer, and this light driven electron transfer leads to photolysis of water, and we have liberation of hydrogen. In addition to it, we can also have indirect biophotolysis, wherein we have oxygenic photosynthesis and hypoxic nitrogen fixation, and both of them are, specially separated from each other. So this is a very unique feature associated with nitrogen fixing blue-green algae, which have developed a segregation or separation, wherein both the processes, one oxygenic photosynthesis and hypoxic nitrogen fixation, both of them are occurring simultaneously. So in indirect biophotolysis, which is much adopted by cyanobacteria, they have specialized organelles called heterocysts. Heterocysts are the structures wherein the cell wall is so thickened that the permeation of oxygen is inhibited. As a result, the enzyme NIF enzyme, which is sensitive to the presence of oxygen, the activity is enhanced. But this unique formula or formulation, or this unique phenomena is observed only in case of nitrogen fixing blue green algae possessing heterocysts. And this phenomena is very well exhibited in nitrogen fixing organisms like nostoc, anabina, calothrix, synicocystis. So these but at the same time, we have certain non-nitrogen fixing genera like Synicocystis and Synicococcus. They also possess this potential of indirect biophotolysis. So we have the pioneer role of enzymes or the biocatalyst, which bring about this whole conversion or production of hydrogen. So we have hydrogenases and nitrogenases. In hydrogenases, we have two classes. One is uptake hydrogenases, which catalyze the consumption of hydrogen produced by nitrogenase, and bidirectional hydrogenase, which have the capacity both to take and produce hydrogen. And nitrogen encoded by the NIF genes, these enzymes are found in the heterocyst of filamentous cyanobacteria, and they grow under nitrogen limiting conditions. They catalyze the production of hydrogen, and and in synergy, they also convert nitrogen to ammonia. So that is the nitrogen fixation, one of the most valuable processes, which is which contributes towards the ecosystem. Had no nitrogen fixation been possible, plants would not have been able to utilize the nitrogen. As a result, the sustainability and the survival for the whole ecosystem is challenging. So hydrogenase gene or hydrogenase was first proposed by Stephenson in 1931 as an enzyme which catalyzes the conversion of hydrogen to protons and electrons, mainly in bacteria, archaea, and eukaryotes. At that time, algal intervention in or the presence of hydrogenase in algae was not explored. So these enzymes, they are specialized to generate energy, and they can generate several other reactions of cellular metabolism. So hydrogenases can be discriminated into three distinct classes based on their active site, the metal which is present at the active site. So we can have nickel iron hydrogenases. Nickel iron hydrogenases are exclusively present in archaea and bacteria. Now archaea are the extremophiles, and the metabolism of archaea is something very unique, which mimics the condition of the earth at the time of evolution, or something around 4,000 billion years back. They still mimic, or they still inhabit such habitats which have a, conditions which are not suitable for the survival of other organisms. So we have the H2 oxidation and production in vivo in case of NIFE hydrogenases. And here the NIFE hydrogenases react with oxygen to give intermediates that can be reactivated. We have FeFe hydrogenases, which are present in bacteria and eukaryotes, which exclusively catalyze hydrogen production, but they are very sensitive, and they can be destroyed quickly, and the reaction can be irreversible. So this is one of the challenges with FeFe hydrogenases. While exclusive Fe hydrogenases are present in methanogenic archaebacteria, which utilize the hydrogen to produce equivalents and different intermediate steps of carbon dioxide utilization to methane. At the same time, they are active. They've got a lot of resilience and they're not at all sensitive to the presence of oxygen. So these are the different figures. We can see NIFE hydrogenase, the structure of NIFE hydrogenase, FeFe hydrogenase, and Fe hydrogenase. These are the schematic structures of different types of hydrogenase, and how they mediated these structures contribute towards their catalytic potential of production of hydrogen. The FeFe hydrogenase and the related hydrogen metabolism genes have been cloned in several species like Chlamydomonas reinhardti and Synodesmus. So the model green algae Chlamydomonas reinhardti encodes two FeFe hydrogenase gene, HYDA1 and HYDA2, which can be expressed only under anoxic conditions. So HYDA1 is the main gene which is responsible for hydrogen production, while HYDA2 contributes towards the secondary role in this, and 68% of the amino acid identity is similar to HYDA, and it plays a secondary role in hydrogen production. While HYDEF and HYDG, they are the accessory genes which encode the protein, which is responsible for the assembly, and it also is responsible for the activation of FeFe hydrogenase enzyme. So, when we talk in terms of hydrogen evolution catalyzed by nitrogenase, uh, uh, we have the reduction of N2 to ammonia, and this enzyme, nitrogenase enzyme, consists of two parts. One of them is denitrogenase, which is also the MOFE protein encoded by the gene NIFD and NIFK, which has two subunits, alpha and beta. And um, and the other is the dihydrogen reductase, the Fe part, the Fe protein encoded by NIFH. These are found in heterocyst of cyanobacteria, photofermentative bacteria, and non-heterocystous nitrogen fixing blue-green algae like Plectonema. And here we can see that this nitrogen, by virtue of the enzyme nitrogenase, gets converted to ammonia, and we have liberation of hydrogen. So we do have these two potential biocatalysts which are bringing about the production of hydrogen. So there's a diversity similar to hydrogenase. There's a diversity in the nitrogenase enzyme, and we do have type 1 containing molybdenum, type 2 nitrogenase containing vandalium, and type 3 containing iron. So these are the several advantages and disadvantages associated with the microalgal hydrogen production. In case of direct photolysis, we can see that ideally it takes 5 to 10 days depending upon the algae species. Some of the algae are slow growing, so they might take time around 10 to 15 days. Some algae require exponential phase to be reached in 10 to 15 days, while some of the fast growing algae they can achieve this exponential phase in 5 to 6 days. So depending upon the type of algae, the direct photolysis would take a range of 5 to 15 days, and the yield also varies from 1.2 milliliter per day to 73.5 milliliter. And through direct photolysis, we have the utilization of water and sunlight. There's no energy requirement or ATP or sugars which are utilized. So that is one of the advantages associated with direct photolysis. The limitation is the yield is relatively low, and we have sensitivity towards the presence of oxygen. In case of indirect photolysis, wherein we have utilization of sugars as well, the duration can last, or the duration range varies from 148 to 168 hours, and the yield varies from 6.62 to 243 milliliters per liter of hydrogen produced. The overall this process has a significant advantage, wherein the limitation or the sensitivity towards the presence of oxygen is inhibited, and the microorganism can be cultured in sulfur deprived media. It can be cultured in industrial feedstock, or we can utilize different inexpensive techniques for their cultivation. So one of the greatest advantages is its resilience towards oxygen sensitivity. But the limitation include the production capability is low, and there are only few algae which have indirect photolysis potential to produce hydrogen. So screening those microalgae or cyanobacteria which have the potential of indirect photolysis, that is a challenging task. When we talk in terms of photofermentation, the time duration is quite less compared to the previous two processes. So here we can have the duration varies from something around 90 to 120, 125 hours. We can get the optimum production, and the yield also ranges from 120 to 140 milliliter per gram. So it is one of the most efficient process compared to other models. High substrate transformation and efficacy is high. We can also utilize different waste products for cultivation of microorganism under photofermentative process. So but the limitation associated with this process is it requires a very large area and specialized anaerobic bioreactors. At times, the yield can also be low. The next option that we have for hydrogen production is the dark fermentation. So, in dark fermentation, we can have the conversion of biomass to biomolecules mediated by bacteria. The, the duration is quite less from say from a few days to it. The duration has come down to 48 to 72 hours, and we can have the yield ranging from 162 to 135 milliliter hydrogen gas produced. So the yield is significantly high. But and another advantage is it doesn't depend upon oxygen. So there's no sensitivity towards the presence of oxygen, and we can have a wide variety or an array of compounds which are produced contaminantly with the with the hydrogen production. So that is another step towards sustainability, and that can also reduce the overall cost. If we have production of valuable co-products associated with the process, so that is yet another advantageous sustainable approach. In addition to it, there are certain limitations associated with this type of dark fermentation. It requires pre-treatment process. Substrate conversion efficiency is low, so it can produce CO2 hydrogen sulfide, which is a limit, which can be a limiting factor in overall production of hydrogen. So these are the different biological mechanisms through which we can have production of hydrogen. And I have discussed what are the limitations with each processes and what are the advantages associated with each processes. So this is the holistic diagram which explains the application of direct and indirect photolysis in biohydrogen production. Now, the major challenges which are faced in producing biohydrogen. So, one of the biggest challenges low light intensity efficiency, and usually we require high intensity of light for this process to occur. Another challenging task is the high cost for product removal. So, when we are designing a hydrogen photobioreactor, the cost economics is a challenge. Another challenge that we face is the lower photochemical efficiency and incompatibility to simultaneously produce hydrogen and oxygen. If we have a co-product formation, that is a way towards sustainability, but in these biohydrogen process mechanism, we have incompatibility to simultaneously produce hydrogen and oxygen. In addition to it, oxygen is a very powerful inhibitor for FeFe hydrogenase. We have enzymatic suppression, and there's also suppression of FeFe hydrogenase gene expression that hinders with the overall yield of hydrogen production. So the inhibition of proton binding required for hydrogen generation is inhibited, which interferes with the hydrogen metabolism in the different microalgal cells, and the overall productivity or the yield of hydrogen is reduced due to presence of oxygen as a inhibitor. In addition to it, we also have mitochondrial respiration occurring in the cell under anaerobic conditions. So, under sulfate derived condition, the protein and the starch content in the microalgal cell it decreases. So, hydrogen evolution cannot be attained continuously. The cell has to you know uh, utilize its metabolic potential for synthesis of essential mitochondrial respiration. So we cannot have uninterrupted hydrogen evolution, which is yet another challenge. So, if we have challenges, what are the strategies which can be used for optimization of the processes or overcoming the challenges? So, one of the options or one of the strategies which has been utilized is elimination of sulfur from Chlamydomonas reinhardti. So, this elimination of sulfur from Chlamydomonas reinhardti has led to a significant effect on reducing the oxygen production rate through direct photolysis. So this can lead to enhance and prolonged hydrogen production. That is one strategy which has been envisaged by the researchers. In addition to it, we have different microalgal mutants which have been generated through genetic engineering, and by virtue of genetic engineering, the availability of glycogen for hydrogen production via indirect photolysis in cyanobacteria can be triggered. So these mutants can address the different challenges which are associated with the hydrogen production. In addition to it, we can also have another strategy, which is the reduction in antenna size. So, optimizing the chlorophyll concentration and the light intensity, we can have a small and truncated light harvesting chlorophyll antenna. So this reduction in the antenna size or truncation in the light harvesting chlorophyll pigment, that leads to an efficient utilization of incident light using a rotating impeller. So that leads to a proper and homogeneous mixing of the culture. And this strategy was used by Ginali in Chlamydomonas reinhardti, and they reported that that leads to a significant yield or increment in hydrogen production. So optimizing chlorophyll concentration, light intensity, and even exploring the different light intensities which increase the production of hydrogen can be a significant factors. We have come across different papers wherein people have used different colored lights, blue light, red light, combination of different lights intensities to evaluate which light intensity, which light color can enhance the production of hydrogen. Similar phenomena we also see in biodiesel production. If we change the intensity of light, some of the studies have shown that blue light triggers the biofuel production. So there can be alterations in the light intensity. There can be alterations in the light quality that can be explored as a strategy towards enhancing the biofuel potential. In addition to it, we can also have nutrient starvation. So some of the microorganism under a nutrient starvation, the productivity of hydrogen is much more compared to the wild type. Like in case of Chlamydomonas reinhardti, the use of magnesium starvation. When the media is depleted of magnesium, that leads to an enhanced hydrogen production. So this is contributed by reduction in the for system 2 activity. And when Chlamydomonas reinhardti is deprived of magnesium, there is a lot of starch accumulation, and this accumulation of starch and reduced PS2 activity leads to a much more efficient utilization of light by Chlamydomonas. As a result, we have higher production of hydrogen. So these strategies have to be worked out individually. I mean, for magnesium starvation can be a significant tool for Chlamydomonas. It might not work very well with Anabena. So depending upon the promising culture that we have screened, all the nutrient optimization strategies have to be envisaged, and there cannot be a universal formula for nutrient starvation for all the cultures. Depending upon their metabolism and metabolic potential, we'll have to envisage the nutrient uh, optimizing conditions. In case of certain organisms or in case of few algae, nitrogen starvation, it also leads to induce hydrogen production. Similarly, if some of the cultures in sulfur derived or sulfur starving conditions, we have enhanced production of hydrogen, while in case of some cultures, we have slower growth rate when sulfur is an inhibitory factor. So once we screen the promising strains for hydrogen production, the optimization, media optimization, and exploring the conditions which enhance the productivity is the second step. In addition to it, we do have a lot of genetic tools which can be utilized for enhanced production. So PS2 sensitivity and PS2 sensitivity towards temperature has been nullified by virtue of genetic manipulation in Chlamydomonas reinhardti, and this had led to an enhanced production of hydrogen. In addition to it, in Anabina, when the hub L gene was knocked out, the hydrogen production it increased 4 to 7 folds in comparison to the wild strains. So, knockout option and CRISPR CRISPR based engineering can be a significant tool for enhanced production. In addition to it, in Synicosystis, it was seen that an oxygen tolerant NIF-Fe hydrogenase can be transferred from other photosynthetic organism into the cyanobacteria, and when this NIF hydrogenase gene was transferred into Synicosystis, which is a non nitrogen fixing cyanobacteria, it showed a great enhancement in the hydrogen production. So, these are a few of the strategies which have been used in case of genetic manipulation that have led to a significant increase in the hydrogen production. In addition to it, the way I was discussing earlier, the light intensity, light duration, light quality can also be a good strategy to increase hydrogen production. So, white light pulses interrupted by long dark phases for sustained hydrogen production has also been reported. In addition to it, we can utilize different immobilization techniques. So, these immobilization techniques lead to enhanced biophotolysis process, and it has been seen that immobilized sulfur deprived chlamydomonas cells within calcium alginate forms have enhanced hydrogen production in even there's a lot of significant research by Professor Anjana Pandey, wherein she has utilized reverse micelles for as a strategy for enhanced hydrogen production. I'll be coming to the details for reverse micelles and other strategies in the next lecture. So, compartment now, how this immobilization technique works. In case of immobilization technique, we have compartmentalization. So, this compartmentalization leads to enhanced production, whether it is hydrogen production or whether it is another biofuel production. Whenever we have compartmentalization, that leads to an enhanced productivity. So, utilizing more sources of immobilization and even co-culturing, a few of the papers report wherein bacteria along with blue green algae or combination of bacteria with algae, and their and this synergistic approach in immobilization. So, co-culturing, immobilization, these strategies can be used individually, or they can be used in isolation for enhanced production of hydrogen. So, this is one picture that gives an overview of all the major approaches in optimizing hydrogen production. So, we have three phases here. One of them is metabolic approaches, the second is genetic approaches, and the third is technological approaches. In case of genetic approaches, as we have been discussing, obtaining mutants which are defective in hydrogen recycling hub SL gene. So that can be one of the great strategies towards enhanced production of hydrogen. Even Hox mutant with oxygen can be one of the good strategies for enhancing hydrogen production. So we have tolerant hydrogenase, the ones which were sensitive towards the presence of oxygen. Once they become resilient or tolerant hydrogenase, they're not at all sensitive towards the presence of oxygen. As a result, the hydrogen production process is much more enhanced. In addition to it, we can also have mutants with lower sensitivity, like molybdenum nitrogenase to ammonia ions. So when this was mutated, it was seen that the hydrogen production is much more higher. We can have pigment reduced mutants. So when it is observed, it is validated that whenever the pigment production is reduced and the mutants which have less production of pigments compared to the wild type, the hydrogen production is much more. Even PAT-S gene mutants, they are responsible for formation and suppression of heterocyst. As a result, we have much more higher concentration of hydrogen. So these are the different genetic mutants which have been utilized, and the productivity has been increased through these genetic tools. When we come to the technological approaches, the technological approaches include improving the technological design of biofermentation of hydrogen producing organism, so which includes immobilization of cyanobacterial cells. It also can it also can utilize fabrication of novel bioreactor system, fabrication of different strategies or novel materials for synthesis, utilizing promising organism or screening promising organisms. So one of the strategy can be screening organisms which are high producers. The second strategy can be optimization of cultivation conditions of the strain of cyanobacteria. So optimization of the cultivation conditions is very significant, because we saw in the previous slide that magnesium deprivation, sulfur deprivation leads to an enhanced production. So until and unless we optimize the cultivation conditions, we'll not be able to fully utilize the potential of the microorganism. And after optimization only we can validate which organism can produce maximum hydrogen under ambient conditions. In addition to it, the technological approaches also utilized use of cheap nutrients and substrate. Now, why cheap nutrients and substrates are always advisable? There are several parameters to it. One of the parameter is, once the source is cheap, the overall cost would be less. That is one strategy. The second strategy is, whenever we are utilizing any industrial waste, whether it is wastewater or multiple wastes which are generated, and when we are growing the microorganism utilizing this waste, it serves dual purpose. One is utilization of waste, that is a sustainable approach. At the same time, utilizing the waste for generation of energy, that is generating wealth from waste. The best strategies, which has been even advocated by the different SDGs. So, immobilization of different microbial cells, optimization of methods for separating hydrogen from impurities of other gases. So, this is also an area wherein different technological approaches are much more required. Post harvest technologies, purification strategies, transportation strategies, storage possibilities. So these challenges can be optimized through rigorous research, and a lot of collaboration is much more required, wherein we have experts from different avenues which can work towards optimization of methods for separation and purification. So drastic strategical approach is required for upstream processing as well as downstream processing, which can lead to a great revolution in the technological approaches. In addition to it, we can also improve the characteristics of photobioreactor. The way I said, in case of upstream processing, we can have much more efficient photobioreactors to increase hydrogen production. Normally, flat panel bioreactors, tubular bioreactors are utilized or vertical column bioreactors are utilized for hydrogen production. So, much more engineering, much more integration of a biologist along with a chemical engineer is the need of the hour, wherein we have fabrication of bioreactors which are low cost. At the same time, the production is much more enhanced. So this was for the technological approaches. Again, for metabolic approaches, we can have utilization of culture conditions. So regulation of external factors. What are the different factors which are influencing the potential, and how these physical factors can be utilized for regulation of hydrogen production. So light intensity, pressure, or presence or absence of oxygen, presence or absence of inhibitors or enhancers, that needs to be explored. In addition to it, we can also use photosystem inhibitors. So whatever processes are not required, or whatever processes can trigger hydrogen production, we can use those photosystems, and we can use the inhibitors uh, judiciously, so that we have higher production of hydrogen. In addition to it, different exogenous electron donors can be explored, so that the potential can be enhanced. And we can create stressful conditions, we can create inhibitory conditions, different limitations can be created, so that the production is enhanced. And anoxic conditions can be created for organisms which perform the best, or the optimum condition or the optimum concentration of hydrogen is produced in under anoxia. So all these strategies require a lot of well articulated experimental design and synergy and collaboration between different experts, so that uh, utilizing hydrogen as a future fuel can be envisaged. These are the recent advancements which show how hydrogen production can be enhanced. This is the first slide with figure that shows that Chlamydomonas reinhardti, it is entrapped in three thin alginate films, and these thin alginate films led to enhanced hydrogen production. We can see the culture of Chlamydomonas reinhardti and how it is entrapped in thin cap2 alginate films and leading to enhanced hydrogen production. The second figure shows that truncated chlorophyll photochemical utilization. So we have the truncated chlorophyll antenna size. Efficient photochemical utilization can be achieved when we have truncated uh, chlorophyll size. In addition to it, we can also use microbial solar cells using mixed microorganism. So utilizing a mixed microorganism has much more potential compared to pure cultures. And this strategy, the use of MFC or microbial fuel cells, can be yet another promising options leading to enhanced production of hydrogen. The application, in addition for to be the fuel, the biologically derived hydrogen can find one perspective is energy. It can lead to generation of electricity, heat, transportation. It can be used, export, and in feedstock. It can be used as a source of ammonia, different chemicals, different petrochemicals, glass manufacturing, metal processing, synthetic fuels, and food. And it can also be used in fuel cells. So there are so many advantageous aspects associated with biologically derived hydrogen. So the future prospects include new research areas integrating hydrogen producing microorganism in mixed culture by direct utilization of algal biomass. So this algal biomass can come from anaerobic digestion. It can come from different nanotechnological or electrochemistry strategies can be used. Immobilization of cells on novel materials, utilization of wastewater resources, and renewable sources of nutrients contributing towards hydrogen production that can be utilized. So combining different advancements in different fields like nanotechnology, downstream processing, or bioprocess technology can lead to a commercially available carbon-free energy carrier utilizing sunlight and water. And in addition to it, application of integrated bioprocesses encompassing wastewater treatment can be a viable solution, contributing towards removal of contaminants from the environment, at the same time utilizing this waste for generation of valuable biohydrogen, a good sustainable solution to the fossil fuels. Thank you so much. And you may please ask the questions that you have.
Thank you so much, ma'am, for such a great presentation. Now this session is open for discussions. Participants, if you have any query, you can ask.
Good
Hello.
morning, ma'am.
Good morning.
Good morning, ma'am. This is Preetha Ghosh with program ID BP 132. My question to you is, what are the technological interventions in biohydrogen production in terms of Indian perspective?
Yeah, that's a good, wonderful question. Thank you so much.
Thank
So,
you.
in terms of the Indian context, we have different research centers. I mean, DBT has established few bioenergy centers. One it is at IICT Mumbai, one is at ICGB Delhi, even IIT Kharagpur. The team by Dr. D Das. You might have listened to his lectures. So there are few leading teams in India which are rigorously working on hydrogen production, and they have brought about a great technological intervention in terms of co-culture, in terms of designing two-step cultivation system, and fabricating new bioreactors. So you can see, I have shared few of the references. Maybe I'll share the presentation with you. There are very good research papers coming from the Indian scientists, which validate the different techno-economical considerations that are being envisaged in India. And I'm really happy that most of the papers are of Indian origin when we see good hydrogen production papers. So you can write to me, or I'll share those papers with you. There are Indian scientists who are working towards troubleshooting the challenges associated with hydrogen production.
Okay, thank you, ma'am. I do have, I think, 2 to 3 questions. Can I ask, ma'am?
Yeah, definitely.
Yeah. And the other question is, as you said while discussing with regard to the biohydrogen production,
Mhm.
you said about the nutrient starvation. So
Right,
that
right.
has yes, ma'am. So that has any link with this limiting nutrient concentration? Means both are the same or with difference, ma'am?
I think more or less both of them represent, or both of them they do work on the similar phenomena, that depriving the microorganism from a specific nutrient condition triggers one or the other metabolic pathway that leads to enhanced production of the biofuel.
Okay, thank you, ma'am. And the third question is, ma'am, with regard to compartmentalization, so which plays, as you said, and which plays an important role, right, in case of this biohydrogen production with regard to the enzymes, localization. Yes, ma'am. So basically, if at all we go through this compartmentalization, in case of eukaryotes, means microbe related this thing, ma'am. In case of eukaryotes, we do have a well demarcated compartmentalization. So now, how that can be interpreted in case of the cyanobacteria, means in what way we can link, right? with regard to the enzymes and its enhance means the enzyme concentration.
In case of cyanobacteria, the nitrogen fixing ones have their own compartmentalization. They have the heterocyst, right?
Yes,
While
yes,
while all other cyanobacteria, which do not have this potential of heterocyst or spatial segregation, genetic engineering, and even these days metabolic engineering and CRISPR-Cas can be an excellent tool wherein we can silence the pathways which are inhibiting towards the production of hydrogen. So CRISPR can be an excellent solution to this problem.
okay, ma'am. Ma'am, and another this thing. So, what are the basic requirements for this biohydrogen production? If at all we want to go in a lab scale. If it, suppose example, I want to go with lab scale production of this particular hydrogen. So, what is the basic instrumentation required, ma'am?
Instrumentation, you will have one session on instrumentation required by Soumya.
Okay,
I
okay.
think on the last day she will get into details for it. But my takeaway message would be screening the promising strains. One of the most significant factor is having strains which are promising, or you establish a process wherein you have co-production. I mean, one blue green algae and one photosynthetic bacteria. So if the organism is good, then you can validate, you can optimize. But until and unless you come up, you have the preliminary analysis with screening of microorganism, that
Yes, yes, ma'am.
taking from A, you can create A to Z, but you need to have that A. Where is that A? The A is a promising strain. So nobody can nullify the potential of microorganisms, right? So we
Yes,
need,
yes, ma'am.
and screening is something very important.
Important.
Sounds very traditional, that screening microorganism in the age of CRISPR-Cas, but yes, this step, no technology can replace this step.
Yes, yes, ma'am. Yeah,
Screening
these
is something, and I always advocate traditional research work. Nothing can replace the traditional work.
the basic bench work, isolation,
Right, right,
screening, and
right,
characterization. Yes, ma'am.
right. So if you have the organism which is promising in production, you can enhance the production. But if the organism yield is not that much, how will you go about creating Z from A which doesn't exist?
Yes, yes, sure. So it's nothing but like microbial consortium, ma'am. The
Right,
co-culture, what you said. Okay, ma'am.
right.
Okay. Thank you so much, ma'am, for answering my queries. And indeed, it was a good presentation. Not good. It was a nice presentation.
Thank you so much.
Thank you.
Any other query?
Thanks, Archana.
Thank you so
Hello.
much, ma'am.
Thank you so much, ma'am. So let's take a break here. The next session will start at 12 p.m.
Okay, Archana, goodbye.
Bye, ma'am.